6 o'clock. Good evening, everyone. I want to welcome all of you to our CDKL5 Town Hall. We appreciate each of you that took the opportunity to send in questions because these submissions were used to shape this conversation. The CDKL5 Town Hall webinar series is an effort that Heidi Grabenstatter, our science director, has been working on to help our community gain a better understanding of the scientific research in the CDKL5 space by gathering experts in the field and having them help break down the science in terms we can all understand. Today, we are joined by Dr. Tim Benke, our lead clinician for our Center of Excellence at Colorado Children's Hospital, and two members of the Ultragenyx team, Dr. Cheryl Fife-Marisich and Dr. Heather Lau. Dr. Fife-Marisich is the Executive Director of Molecular and Cell Biology, who leads a research team of scientists and associates focusing their efforts on the company's preclinical neuroscience programs, including an AAV9 gene therapy for CDD. Dr. Heather Lau is the Executive Director of Global Clinical Development, who recently joined Ultragenyx to lead the clinical neurology program development in CDD. Dr. Lau is a board-certified neurologist who for the past 10 years ran a large multidisciplinary clinic and clinical research center at NYU that was dedicated to diagnosing and treating patients with rare neurogenetic diseases. The full bios for everyone participating today can be read at the link provided in the email received with the webinar information. Recently, we were able to hear a presentation from Dr. Fife-Marisich through the CDKL5 Alliance International Family Conference that was held in June. This evening, we are hosting an extension of this conversation, including Dr. Benke and the Ultragenyx team. As parents, it is hard for us to sort through the science, especially understanding the research done in models and how this research will lead to possible treatments and benefit our loved ones. We hope this conversation will help us all have a better understanding of some of the challenges as well as the steps needed to take bench science to bedside treatments. And with that, I'm going to now turn it over to Dr. Benke.
Thank you, everybody. I'm going to share my screen so you can look at my slides. There, you should probably be able to hear me and see my slides now, hopefully. So thanks everybody for taking some time to join us. So my name is Tim Benke. I am a pediatric neurologist and I'm at Children's Hospital Colorado and the University of Colorado. I am medical director of the Rett Clinic where we see individuals with CDKL5. So these are my disclosures, and I think it's relevant that I note that there are some advisory that I do for both Ultragenyx as well as another company called Alcyone. So clinical trials in CDD, here we are. So this is where we want to be for treatment specific to CDD. And how we have to get there is through both preclinical research and then research through the clinical trial process. And it's important to remember that where we're at right now, this is still research and not treatment. So ideally, a successful trial will lead to FDA approval, and then there will be availability of treatment for everyone. So what's your role? Your role is, as Karen was saying, ask questions, get informed. If there's a clinical trial, please enroll, but also be patient, please, with the process. And that's part of what the reason for this talk is to let you know what the process is and why it takes so long. So bear with us. So here's one of the questions that comes up is, what do I do all day? Well, it's not the McDonald's model, where McDonald's can sometimes have a few people in line, or they can also go to a lot of people in line. I've always got a lot of people in line with a lot of clinical duties with Rett Clinic, General Neurology Clinic, Neurogenetics Clinic, clinical trial patients, and on-call service. I'm also involved in research grants, one of which is the U01 study to investigate outcome measures in CDD and biomarkers, and I'm program director for that. And then I'm also involved as principal investigator on a study with NIH to investigate inhibitors of the CDKL5 protein. I'm also involved in other grants from other people like Simon's Foundation and other NIH grants. I also have a lot of administrative work to do. So, clinical trial basics. So what is a clinical study or a clinical trial? So it's a process by which subjects or caregivers consent or assent to participate in a defined study designed to produce information that can be used to understand or study or help, as in a trial, the population as a whole. This data is then peer-reviewed and published to advance knowledge and effective and safe treatments of the disease. Trials are interventions. This is all voluntary. You do not have to participate in a clinical trial unless you want to participate. Participating involves consent. In some cases, using de-identified data, you opt out versus opt in, and you can withdraw at any time. A review board must approve the trial before any subjects are even contacted, and then following that enrolled. And there are international rules of standards and ethics that we have to follow. So there are phases to a clinical trial. Phase 1 often tests healthy volunteers at multiple doses of a drug. It may be in patients with advanced stages of a disease or diseases with no known treatments. Safety is primary in phase 1 trials. Phase 2 is testing on patients for efficacy and safety. And phase 3 is a confirmatory trial. It's testing on patients for efficacy, effectiveness, and long-term safety. The FDA will typically approve for use after a phase 3. This may be different or exceptional with gene therapy because this is new territory. So there are a lot of different trial types. So placebo controlled. So a placebo, also called a sugar pill, is used to make sure the effects are real. So patients with epilepsy can see a 25% reduction in seizures with a placebo. And so that's why it's important, even in epilepsy, that a placebo arm is there. Randomized, that's who gets placebo or drug as a flip of the coin. Double blind, this means subjects and the local study personnel, including myself, do not know who is getting a drug. Open label, that means everybody's getting the drug. Crossover, this means those that were getting drug then get placebo and vice versa. It's also usually randomized and double blind too. Extension, this is access to a drug after a trial ends, and usually an open label continuation of the trial. So the gold standard is randomized, double-blind, placebo-controlled, but we don't know if this is what the FDA is going to want or will be true for gene therapy trials. So clinical trials. So what should you expect as a participant? And it's important to remember that we live and have to follow the protocol. The protocol sets up everything that we're allowed to do in the study. We can't go outside what's in the protocol. So the first thing it sets up is something called a screening visit, which is the first visit to make sure that subjects meet the inclusion and exclusion criteria. Then consent or assent is signed. And it's often a lottery, and usually the industry approves who the participants are. So I don't necessarily get to decide who participates in a trial. It's sometimes the industry, because they're looking for certain patients at certain sites. The inclusion and exclusion criteria, it's usually something like age, weight, disease specifics, other medication use, recent trial experience, etc. And there's no exceptions to this. It's written in the stone of the protocol. The duration, this is the duration of treatment and follow-up during the trial, and study visits. So these are specifically timed for the trial to get good data. They're not that flexible. It's usually plus or minus a day, not weeks, and it's not optional. The study coordinator will work with the families to try to fit it into their schedule, and they happen over the duration of the trial. Family friendly is considered as much as possible. Endpoints or outcomes. These are the measurables to determine safety and efficacy. Clean data. So this has to do with everything about the exclusion and exclusion criteria, and we need to be sure that whatever we're doing is safe and effective. And this is research, it's not treatment. Post-approval, regardless of inclusion and exclusion, hopefully this will be available to all. Good data. Another piece of this is that it's important to have the same caregiver present at each of the study visits to ensure that we're collecting consistent data. Also, there are travel issues. Travel can impact how somebody behaves, and so sometimes travel is not allowed with some studies. Clinical trials. Patient voice. This is important because what the structure of the trial looks like this is important to the FDA, and so they will often ask you, the families, so what types of trials are acceptable to you? What types of symptoms should be treated? How are these symptoms assessed? What are meaningful changes in those symptoms to the family? How does the age of the individual impact all of these questions? So the parent voice and FDA interactions usually happens through something called a patient-focused drug development meeting. And this happened for CDD already in November of 2019. And there is a summary at that website there. You can also Google it and you should be able to find it. Capturing the outcomes correctly is really important. So this started for us with the NIH-sponsored U54 study to determine natural history. And as part of that, we came up with the first CDD-specific outcome measure, and we are studying this and other measures further in the NIH-sponsored U01 study titled Development of Outcome Measures and Biomarkers for CDD. There's also the Lulu-sponsored CANDID study, which is studying previously established outcome measures, though not necessarily specifics for CDD. Both of these studies are very important to decide what are the right outcome measures to use. So what are the top concerns in CDD? So from the NIH and NINDIS natural history study, so this came from Jeff Newell, the top 3 concerns were seizures, communication, and sleep. And from the FDA PFDD, similar list of issues. The first was global developmental delays, second was seizures, and the third was GI issues. So this is important. So why aren't we doing anything? Why does it take so long to get started? So the first step is getting preclinical data. And Cheryl's going to talk to you more about this with her talk. But basically, it's to determine the efficacy and safety in animal models. And this happens both in the academic side of things as well as in industry. And that's also one of those things that's going on is what we're doing, as I was talking about, the outcome measure development. And then there's the solicitation of parent voice, and this is often done by industry interacting with families and with the FDA. There's human protocol development. This happens usually with industry, sometimes with input from some of the sites that could be involved. And then there's federal regulatory approval. So the company then files something called an investigational new drug application, or IND. And that's what kind of starts the process going to the next step. But this whole thing takes about 5 to 10 plus years. If you look at what's going on with Rett, you can kind of map that out, and it's been over 10 plus years. Next thing that happens is site recruitment and approvals. Then there's contract and budget approvals with the sites. The sites then make sure that they get ethical board approval. And then there's this thing called a site initiation visit, where all of the sites are trained up to make sure that they know how to administer and do everything the way that it's outlined in the protocol. And then finally, enrollment can begin. And we call you, you show up, and then the trial starts. And that process starts about 4 to 12 months. So we are here. We're at numbers 1 and 2. So clinical trials, where do you get information about all of this? Facebook posts have contaminated trials in the past. So don't be that guy that ruins a trial for the rest of us. Email, internet, blogs, me. Well, I do not endorse any particular trial, though I will participate in some, and I can be a source of information if I'm allowed to provide it to you. But really, trust but verify. The only real sources are clinicaltrials.gov, the company website, the consent form of the study, or the study investigators, so I can tell you about the trial after it's IRB approved. So I want to back up and just make sure that we're all on the same page about some key concepts. So I wanted to talk briefly about genes and proteins. So a gene is a segment of DNA that codes for a protein. Proteins are made up of smaller units called amino acids. There's 20 different types of amino acids. And the sequence of amino acids determines each protein's 3-dimensional structure and therefore their specific function. A protein is a molecule that can have many different important roles in the body. They provide structure and function, transport materials, send signals, and they protect the body from viruses and bacteria, and they carry out chemical reactions. So the gene is like the recipe. The amino acids are like your ingredients. and your proteins are your cake that you end up with. So that's arranged in our body in little units called cells, and there's a certain part of the cell called the nucleus, which is where the chromosomes are, and the genes live on chromosomes. And those genes, as I said, are the instructions on how to make proteins. So there are different types of gene therapy, and they fall into different categories or strategies. So the first is gene replacement, and that's what I'm going to talk to you primarily about today. There's other things such as gene editing, protein replacement, and also gene read-through. But like I said, we're just going to talk about gene replacement. So what are the issues? So gene replacement. So there is some excitement here because first of all, findings in animal models. So an artificial turn off and then turn on again in CDD mouse models, which really mimics the ideal situation where there's perfect gene replacement therapy, has shown promise. The animals appear to get better. And then gene replacement studies in CDD mouse models, and then also gene replacement in other disease models such as Rett, spinal muscular atrophy. And then what's really got everybody excited is findings in humans. So there's been dramatic response in a disease called spinal muscular atrophy. Spinal muscular atrophy is a catastrophic neuromuscular disease in babies. It's catastrophic because those individuals die by the time they're 2. And with this gene replacement therapy, not only are the babies alive, but whereas normally they were too weak to breathe even, these babies are able to sit and walk. So it's dramatic. So what are some of the issues? So this is an addition of a new copy of a gene. It's not really replacement, we call it. So it's suitable for any loss of function alteration of CDKL5, which is what most of the different changes in CDKL5 are that we see in CDD. So some diseases are what we call Goldilocks diseases. So it's not too much, not too little, but just right. So in some of those disorders, it's important not to overshoot and have too much replacement. But in general, we think that you want to replace only just enough in any disease, because if you overshoot, that might actually be toxic. So you want to know how to get the replacement into as many cells as possible and how to prevent any possible immune response to this and to prevent any other side effects. So gene replacement is done through something called adeno-associated virus, or AAV, and it's the viral vector of choice. And that viral vector is used as a taxi to deliver the gene. And I've got my little cartoon down there in the bottom of a local taxi that's filled with an awful lot of people. And this is usually what you're doing with your vector, is you're filling it as full as you possibly can. So the viral vector, the virus, is re-engineered to deliver the gene of choice and regulate how much replacement protein is made. So it does not make any more virus because the virus has been rendered replication incompetent. Natural AAV is not known to cause any diseases in humans. And it's a viral vector because we need it to cross the blood-brain barrier in some situations. But it's important, like the taxi, the virus can only hold so much DNA, so you're limited to the size of the gene. But fortunately, the CDKL5 gene will fit in there. So what are some of the issues and questions? So the first is intravenous versus direct CNS delivery. So we think that there's going to be a less immune response if it's direct, but that's going to be more invasive. Direct, we think that there's going to be fewer systemic side effects, such as with liver or blood irritation. We know from the SMA studies that those babies, when they got the gene replacement therapy for SMA, it was delivered via the blood. And there's both an immune response, and there's also sometimes irritation to the blood or the liver. How many cells in the body need to be altered to have a meaningful change? Well, 10 to 25% may be the most achievable currently. But as some of the things that is going to be in Dr. Fife-Marisich's talk is that may be enough. So the immune response. So this is something that the body hasn't seen before in terms of individuals have usually not seen adeno-associated virus. So there is an immune response associated with that. And we know through the use with in spinal muscular atrophy is that immunosuppression is necessary. And this has to happen for weeks to months. And that's important because this is a one-shot opportunity. Because if the body already has or develops an immune response to the AAV, it limits its effectiveness. So repeat dosing is not an option because of that immune response. It really is a one-shot opportunity. It's important to note that the gene does not insert itself into the rest of the chromosomes. It just sits there. It sits in the nucleus of the cell, and it's unknown, but we think it just stays there for forever in the case of neurons. But we don't know if it stays forever, but we think it does. And it's important to note that germline, that is egg and sperm, are not corrected. So they're not affected by this. So the gene replacement does not correct the egg and sperm. So what's the optimal delivery route? So global delivery is, I hope you can see my cursor here, would be with intravenous. So it's easiest, but it does require higher doses, and it may not target the brain, and it does have the highest chance of eliciting unwanted liver, blood, and an immune response. So delivery via the CNS, so there's a couple of ways to do this. This can either be intrathecal in the lumbar region, or intrathecal in the cisternal region, or intracerebroventricular. That is, you're going to have to go through the brain. So these methods, they're potentially challenging to deliver, may not reach the deep brain structures, but will target a higher percentage of neurons compared to IV at a similar dose. To do this intracerebroventricular route, as you can imagine, it's more invasive because you have to go through the brain to get there. Another possible way is to do injecting directly into the brain, which would be called intraparenchymal delivery. And this potentially targets a large percent of neurons, but only in those select areas where you inject the drug. So in clinical trials, in spinal muscular atrophy, the drug for that in gene replacement therapy, it's got a long name. It's called onasamnogene albupervivac, and the FDA has now approved it. And it uses a very similar vector, very similar AAV as that which is proposed to be used for gene replacement trials in CDD. So general unknowns. So these are questions that are for both you as well as me. So how much improvement is enough for you to participate in this? How long is it going to take to see an effect? And this is important. We don't know the answer to this, but this influences how long the trial duration may be. It's anybody's guess, but at least in terms of the trials for SMA, those trials lasted years. What are the long-term consequences of these therapies are unknown, and that's another reason for having trials that extend over several years. Is there a critical window of opportunity for treatment or not? There are some recent studies in animal models that suggest that there may not be a too late to pursue this. Another important thing is how to pay for this once approved. Onasamnogene albupervivac is the most expensive drug ever approved. It's $2.1 million just for the drug. And it doesn't include everything else, such as the anesthesia and immunosuppression, et cetera. So in summary, gene therapy is viable, and many disorders, previously catastrophic ones like spinal muscular atrophy, may have therapy. A lot of these unknowns will need to be addressed, but many will only be explored through the clinical trial process. So thanks, everybody. I really appreciate your attention, and there are some other resources, but look forward to if there's any other questions. But I did try to answer all those questions that showed up online. So now I'll turn it over to the Ultragenyx team, Drs. Lau and Fife Marisic. Let me stop sharing. Oh, thank you. I think you kicked me out.
Hi there, good evening. Once again, this is our the Ultragenyx group. So we'll have an update from us on the status of our AAV9 SYNH CDKL5 program. And we'll start off with a little bit about Ultragenyx pharmaceutical company. We'll talk a little bit about the cross-functional approach to better understanding CDD and how it affects our patients and families. And finally, Dr. Cheryl Fife-Marisich will talk about our translational research program for CDD. So as we were introduced earlier, I'm Heather Lau. I'm the Executive Director of Global Clinical Development, and I'm a child neurologist by training. And as you can read about, I've been a clinician for the past 10 years and partnering with my families that I've taken care of over the years to help them understand rare disease research and understanding the whole process of going from bench to bedside with these clinical development programs. So a little bit about Ultragenyx. So Ultragenyx is actually a biopharmaceutical company that's been committed to advancing life-changing treatments, providing access to those in need, and supporting the larger rare disease community. It was actually founded by Emil Kakkis in 2010. He had begun his work in developing enzyme replacement therapy for a rare disorder called mucopolysaccharidosis type 1, or Hurler's disease, in partnership with the Ryan Foundation. I've known Dr. Kakkis since 2012 and watched this company grow from a small startup of less than 50 employees to now more than 1,000 employees. It's headquartered in California, and now we have global offices throughout Europe and Latin America, and a presence in Canada. So at Ultragenyx, our mission is to rapidly transform undiscovered discoveries into effective rare disease products. And how do we do this? So we focus on the serious diseases with clear biology. We then remain agnostic to modality, relying on that clear disease biology to identify the best path forward. What does that mean? That means we're not tied to either small molecule or protein-based therapies or gene therapy. We look at the disease itself and understand it better, and then decide, how do we approach it? How can we develop disease-modifying therapy? We remain focused on these essential questions and try to start to incorporate that pathophysiology of that disease state, and then lead to understanding, how does that impact the actual patient and their caregivers? And we involve clinicians very early on to gain a better understanding of disease pathophysiology. And we leverage our animal models to further understand this disease. And throughout this entire process, we partner with our patient communities to ensure that our patients and caregivers' insights are consistently incorporated throughout the development of this therapy. So what is Ultragenyx doing today to help us move closer to conducting a future potential trial for therapy for CDKL5 deficiency? So we are starting to advance research in partnership with Allstripes. Allstripes, which you might know and contributed to, is a research study that's collating medical records from our patients with CDKL5 and then de-identifying it. The study aims to better understand our patients living with CDKL5 deficiency and learn how these patients are interacting with and using the healthcare system. So for example, how are they diagnosed? How long did it take to get diagnosed? What types of doctors are they visiting? What types of therapies and treatments are they using for their management? What aspects of healthcare are being utilized? We're also collaborating to support the natural history studies. As you heard from Dr. Benke earlier, there are several studies out there that are going on. And we are one of 7 companies supporting that 3-year observational clinical assessment of neurodevelopmental measures in CDD. This is called the CANDID study that's led by the Lulu Foundation, looking at various assessments, validated assessments to see how it changes and is impacted by the various symptoms of CDKL5. So impact on motor, language, sleep, behavior, and seizures. We are also an invited member of the External Advisory Committee for the NIH and NINDS funded U01 grant that's looking to understand and validate biomarkers and core clinical features to prepare for clinical trial readiness. And this is ongoing as well. But most importantly, we're learning from our caregivers and clinicians. And we're doing this through conducting one-to-one interviews with our caregivers and healthcare providers who are experts in this field to build on the existing research to understand the impact of CDD on patients. This is hoping to advance our global conceptual model of CDKL5 so that we understand the entire impact of this disease, including its impact on the physical symptoms, the social and emotional impacts from day-to-day living. We also are meeting with advocates, most recently at the CDKL5 Forum this past month, to discuss the challenges and opportunities related to biobanking and the role in drug development. What is biobanking? Why is it important? What are biomarkers? Why are they important in the overall clinical trial development plan? Furthermore, we're hosting insight meetings with panels of caregivers. So, the first stage was to learn how this disease impacts a child in all domains. But now we're trying to understand what are the treatment goals that are guiding your decisions about the care and management of your child. What are these key decision factors when you're seeking care and treatment? And more importantly, for you yourself is understanding what is gene therapy, how is it impact, what are the implications of gene therapy, and what does it mean to participate in a clinical trial. And finally, we created a Ultragenyx-driven gene therapy advisory council, and we're partnering with patient advocates to understand not just with CDKL5, but other disease states, including Wilson's disease, OTC deficiency, glycogen storage type 1A, Duchenne muscular dystrophy, and spinal muscular atrophy. We're bringing thought leaders together to understand the challenges across all different types of disease states when we're embarking on gene therapy. We need to understand the challenges, the knowledge gaps, and the unmet needs as we embark on this new type of therapy. So these are the ways that Ultragenyx is focusing on developing our CDD program while we're still making advancements in our preclinical program. And so I will turn this over to Dr. Fife Maricic, who will now discuss our preclinical program in CDKL5.
Great, thanks, Heather. So I know a number of you, a little bit of background. Heather, maybe there we go. Not sure if that's why it was. So I know a number of you have been asking what a typical day might look like for those of us who are doing translational research. So I put together this slide to sort of help me describe this for all of you. So a typical day looks basically like these pictures here on the left, at least before COVID, and we're hopefully making our way back to this. The picture on the top is the team that works directly with me, and you can see I'm in one of those pictures. The picture on the bottom is an older picture of the larger translational research group. We've actually grown quite a bit since this picture was taken, but we haven't had a chance to be all together without masks in quite some time. So this is taken pre-COVID times. But basically my day consists of overseeing an amazing group of really talented scientists with expertise in molecular biology, neuroscience, and cell sciences who work on many different rare disease programs in addition to CDD. So we work on liver diseases, bone diseases, many things, and in addition to different neurological diseases, including CDD. We conduct regular group meetings. We communicate a lot and share our learnings. We have project-specific meetings as well as one-on-one meetings where I can help a lot of my team with their career development and other things like that. And then I also oversee, probably this last point is most important to all of you, I oversee the non-clinical or the preclinical subteam of our CDD program. And that's composed of scientists who specialize in many different areas of drug development. And there are about 20 of us on that subteam. And then there are many different subteams, including a team led by Dr. Lau, who focuses on the clinical aspects. And we have people who focus on the manufacturing aspects. And I'll talk about that in the next couple slides. All right, so next slide. Looks like there might be, oh, there we go, thank you. All right, so one challenge in the development of a CNS gene therapy is the scale up from a small mouse brain to a much larger human brain. And I included the pictures on the left of this slide to just really try to illustrate this for you. Our mouse models are really powerful, and I'm happy that we have these available to us to use. They're really good for helping us understand how our gene therapy vectors function in the context of the disease. So this is where we can look at potential efficacy. And the reason why we use rodents for this is it's fairly easy to manipulate their genome. So we can create disease models, and then we can look at their behaviors and see if they mimic any of those that are found in patients. The problem with the mouse models is, of course, a mouse brain is not a human brain, right? The rodent models, that the brains are much simpler, and they're much smaller. And that's a problem for treatments like gene therapy, where we really want to have a really broad distribution of that gene therapy vector across the brain. That's pretty easy to do in a tiny mouse brain, much more difficult to do in a larger human brain. And so we also use monkeys with much larger brains to try and get at this question of looking at biodistribution. What does it look like in a bigger brain? But the monkeys are not typically disease models. So there are some monkeys now that have naturally occurring mutations, but there aren't any that I'm aware of for CDD, and there are very few true disease models in the monkeys. So the monkeys are healthy monkeys that we use. So they're more useful for understanding safety, and also for, as I mentioned, optimizing that distribution in a larger brain. So as the Ultragenyx CDD program aims to eventually move from mice into humans, we are also thinking about how we are going to be able to produce enough vector. And this is another problem that we see that has to do with the scaling, right? We can make vector to treat the mice for our preclinical studies. But as we start thinking about how much vector we would need, probably about 1,000 times more to treat a human being, we have to think about how we might do that. And so what is Ultragenyx doing? How are we working to ensure that we can make enough vector to treat patients with CDD? Next slide. Well, we can take advantage of our proprietary HeLa platform. And as I just mentioned, one of the major bottlenecks in developing a gene therapy is the scale up of this manufacturing process. So our proprietary HeLa producer cell line, or our PCL platform, we hope will enable us to scale up the manufacturing process in an efficient way so that sufficient drug product can be made to serve all eligible CDD patients. And we are really committed to this, and we are developing a 100,000 square foot manufacturing facility in Bedford, Massachusetts, slated for completion in 2023. And we really believe that this will provide us with the flexibility and the control of the process to further optimize our HeLa PCL platform and really enable us to produce enough gene therapy vector to treat large patient populations. So we're very excited about this, and we wanted you to know that we're thinking ahead. We have a lot of work to do. We're still early on, but it's important to start thinking about these things before it's too late. Okay, for the next couple slides, I'm going to go through some slides that you have seen if you've watched my CDKL5 Alliance talk, and just take a few moments to go a little bit deeper and explain things a little bit more based on some of the questions that you've all asked. So here on this slide, you can see our investigational gene therapy product for CDD. And the therapeutic approach here is to use a single injection of the vector to the cerebrospinal fluid. And the aim here, or our goal, is to supply a functional copy of CDKL5 to CNS neurons. So on the very top, you can see this is the DNA that we package into that AAV9 capsid. And you heard from Dr. Benke earlier today in depth about how this works. So we have a sequence of DNA that encodes the synapsin promoter. And what that does is allows us to drive expression of CDKL5 specifically in neurons. And then we have our human CDKL5 DNA sequence. And then we package all of that into the AAV9 capsid. That is then delivered into the cerebrospinal fluid. You can see that shown in blue. And that enables our vector to travel throughout the brain, where it can travel throughout the ventricular system, but then diffuse out into the brain parenchyma. And that's shown in gray. And that's where it can come in contact with neurons. It can be endocytosed or taken up by that neuron, it can travel in endosomes. Then it is released, makes its way into the nucleus, where that capsid can then be shed, right? The capsid is that taxi that Dr. Benke showed. The capsid is no longer needed, it's shed, and the DNA is deposited in the nucleus where it does not incorporate, and it exists as an episome. And then the host cell machinery can take over and then produce the protein. So the goal here is to supply the cell with functional CDKL5 protein. So on this slide, what I wanted to do is try to convey sort of how complicated it is when we're in the preclinical space. And you got a taste from Dr. Benke's presentation about the whole process, right? And we're still very early in that process. But even that early stage has many, many points that we go through. And these are just some of the things that we think about on a daily basis. So there are several fundamental questions that we need to answer in the preclinical space here. The first is, what is the appropriate therapeutic transgene? What is that sequence of DNA? Exactly what does that look like? What do we need to build the vector? The second is, where and what cell types do we need to target? The third is, how many cells do we need to target? And when in development do we need to treat? And I have that highlighted because I'll go through that a little bit in the next slide. We also think about how much vector-derived protein we need to express in order to achieve efficacy. So even when you get that to the cell, do you need to express a little bit of protein, or does that cell need a lot of protein? And that's an important question. Number 5, another very important question, is this treatment safe? And we spend a lot of time on this one, and there are a lot of regulations that we need to follow to make sure this is safe before we even think about designing a clinical trial. And the 6th is, are there any fluid or functional biomarkers that could help predict clinical benefit? And this can be really complicated, particularly for a CNS disorder, right? We can't sample a little piece of somebody's brain to see if it's working on a molecular level. We can do that in the mice. We can't do that in people. So we have to think about, is there something in the blood that we could follow? Is there something in the cerebrospinal fluid? And so we're working really hard to try to get to the bottom of that. All right, so this slide describes our mouse model study design for one of our key mouse model studies that were done in collaboration with Dr. Rodney Samako at Baylor College of Medicine. And it's a busy slide, but there are a couple key things to pay attention to. The first one is the age where we decided to treat the mice. And we took a slightly different approach to what you often will see when people are testing gene therapies for CNS disorders preclinically. Most of the time, you'll see that people inject mice right when they're born, early postnatal. The problem with that is that that translates to a 3rd trimester fetus in human. And we know that currently we're not able to treat that early. And so we wanted to really inject these mice at a time point that would be translatable. And so at 4 to 5 weeks of age in a mouse is roughly equivalent to 4 to 11 years of age in human brain development. The other reason why we wanted to inject the mice at this age is because we know that as the mice get older, it's much more difficult for AAV9 to travel and distribute really nicely throughout the brain. So when you inject the mice when they're first born, you can get about 60 or 70% of neurons hit with your vector. So it's very, very high. As the mice age, that number goes down. And at 4 to 5 weeks of age, we saw that we were able to transduce or introduce our vector into about 10 to 20% of neurons. And that's much more similar to what we see we can achieve in a larger brain. So we wanted to know whether if we only hit a small number of neurons, is that still beneficial? Will that still help improve some of the behaviors that we see in the mouse model? So after we injected the mice, and we did inject them into the lateral ventricle. So we went right into the cerebral spinal fluid with what we call an ICV injection. And then we waited until the mice were adults. So 10 to 12 weeks of age in males, or 16 to 18 weeks of age in females. And we wait a long time on purpose because we wanted to make sure the brain had time to recover. Once we've introduced a functional copy of CDKL5, maybe it needs to rewire a little bit. We wanted to make sure we didn't go too early. And the other thing we did in this study was we looked at both males and females. And we thought this was important because the biology is different. Because CDKL5 is on the X chromosome, in females, we all have 2 copies of the X chromosome. But in males, there's only 1 copy, 1 X and 1 Y. So to control for that dosage, females have something called X chromosome inactivation, where you're only expressing from 1 of your 2 X chromosomes in every cell. So what that means for females with CDKL5 deficiency is that half of their neurons have CDKL5 that's functional and the other half don't. Whereas in males, unless you're mosaic, none of the neurons will have functional CDKL5. And so long story short, we saw a pretty similar response from the males and the females to our treatment, which was a little bit surprising to us. They both seemed to do well. And so we were able to pool that data together when we looked at the results. So on the next slide, I've put everything into just a quick summary, so I don't have time to go through it, but if you do want to watch that CDKL5 Alliance talk, I do go through some of the behaviors that we looked at. But I'm using this slide to show you that I think that there is reason for hope. We were able to see improvement across many different behavioral assays. So this is good, but we didn't completely fix it. And I want to be 100% honest about that. We were targeting a small number of neurons, and we were injecting into mice that were a little bit older, right? So it's amazing that we were able to see the improvements that we did, but they're improvements. They're not cures. There was only one assay where we completely restored the learning and memory behavior to normal. In the other cases for motor function and for some other learning and memory assays, they were improved. And we think this is a good thing. And then there were some other things, and you can see it denoted by the red X, where we didn't fix it. And so I think that it's important to be hopeful and to feel positive about this, but to remember that this is a mouse model, right? And so we don't know how this will translate to people. We're hopeful that it will translate, but that there are improvements across many domains, but not everything. So the next thing I'll go through quickly is a set of studies we did in collaboration with Allison Motry at UCSD School of Medicine. And Dr. Motry had shown in his lab that he saw a hyperexcitability phenotype in neurons that came from patients with CDD. And the reason why we wanted to partner with Dr. Motry here was because, as I said, the rodent models are great and they're very powerful, but they're rodents, right? And so it was important for us to look to see what might our gene therapy look like if we were treating human neurons. Now, of course, these assays have issues as well because we can't look yet. We're still preclinical, so we're not looking in a human brain. We're looking in a dish, but they are human neurons. And so I think everything has, both types of assays have their pluses and minuses. And I think it's powerful when you look at all of the data together. So here, what Dr. Motry had done was taken skin cells from patients and created induced pluripotent stem cells that he then differentiated into neural progenitor cells, and then differentiated further into cortical organoids. What these are kind of like miniature human brains. It's not exactly that way because it doesn't completely model the entire brain. This is modeling the cortex. That's why we call it the cortical organoids. But it does a pretty good job of forming the different layers of the cortex, and it's a little ball or a little mini brain that grows in a dish. And we can grow those on a set of electrodes that can measure their electrical activity. So the way that neurons communicate throughout the brain is through firing of action potentials. We can measure that as electrical energy. And we can show that at baseline, the the cortical organoids from CDD patients are hyperactive. They fire too many action potentials. And then we can look to see what happens after treatment with our AAV gene therapy. So on the next slide, on the very left, you can see here we added a green fluorescent protein tag to our vector so that we could show and count the number of neurons that took it up. And we could see that with about 15% of cells that were targeted, we were able to see some pretty dramatic improvements. So on the right-hand side, where you look at the graphs, the gray dots show you the mean firing rate, or the number of bursts per minute in the cortical organoids from patients with CDD. You can see that they're hyper excitable. Their mean firing rate is too high, and the number of bursts per minute is too high. 2 weeks after we treat with our AAV9 gene therapy, we can bring this back down to the levels of the control. So the message here is that transduction of a small percentage of neurons leads to functional improvements in the cortical organoid hyperexcitability, which is similar to what we saw in our mouse model. We saw a small percentage of neurons was leading to improvements in motor behavior and cognition. And here we're using a different system and seeing something similar. So I think that there's reason for hope. I think these are positive data, but they are only models, right? And we don't know how this will translate into people. And we still have a lot of work left to do before we're ready to get to that stage. I think this is my last slide. I think we have time. I'll go through it quickly so we can get on to the questions. I just put together a few bullet points to outline some keys to developing a successful AAV9 gene therapy for CDD. The first is really to work to develop a deep understanding of the full phenotypic spectrum, as well as the underlying biology of CDD. We need to understand this disease as well as we possibly can, because that helps us develop reliable preclinical models and robust biomarkers. We need to use our mouse models for what they model best, and not try to force things that they don't. At the end of the day, they are rodents. And so there are many things that they do model well, and we can use those. But for other things, we can turn to other models, such as the human iPS-derived neurons that I talked about, so that we can better understand additional aspects of the disease. We need to determine the optimal immunosuppression protocols, and Dr. Benke talked a little bit about this today already. We understand from other gene therapy programs that the immune system does react to AAV, so we need to make sure that we're on top of that. And we also need to work on determining the optimal delivery parameters so that we can maximize the number of neurons that take up our vector across the brain, particularly in larger brains like monkeys and eventually in people. And then we need to develop appropriate clinical outcome measures and robust objective biomarkers that can capture key aspects of the disease and how those aspects change over time. And that's something that Dr. Lau and her team are working really hard on. And that, I think, is it for me. I think we're going to move on to some questions. Thank you.
Thank you to all of our speakers. You've provided a very comprehensive roadmap for where we are and where we still have a lot of work to do, both preclinically and clinically. We sort of have a roadmap in front of us of the work that is yet to be done. I'm amazed at how much progress has been made using the viral vectors in mice. I really appreciate the job you've done incorporating all the questions that came in from our caregivers in your presentations, but we have a few more that remain. Forgive me if we repeat ourselves from the presentations, but I don't think it ever hurts to hear things more than once as kind of on our trajectory of learning. Dr. Benke, one question asked, will patients who have a complete gene deletion benefit from gene therapy?
Yes, we think that anybody with a loss of CDKL5, which would include complete gene deletions, would benefit from gene replacement therapy.
Fantastic. And Dr. Five Marisic, will there be an age restriction that you anticipate?
Thanks. So Ultragenyx still has a significant amount of preclinical research and work ahead of us, as I was just mentioning, before we can really determine if it's possible to conduct a clinical trial in patients. So I don't know the answer just yet, but we have thought about this question. And although we don't know for sure how well older patients might respond to gene therapy for CDKL5, we did design our mouse studies, as you just heard, right, to try to better understand whether treatment with AAV9 at older ages could be beneficial. And so because we treated those mice at a time point that corresponds to about 4 to 11 years of age in human, and we saw improvements in behavior, we're hopeful. There's also a new study that was published out of Joe Zhao's lab at UPenn recently. This was in the last couple of weeks, where he used different genetic techniques, and Dr. Benke talked about this, to restore CDKL5, which would sort of be the gold standard. And he did that in mice that were equivalent to teenage years in human. And he saw that there were benefits to that. So I think that there are a number of studies now that are starting to point to the fact that there should still be benefit even in older patients, but we won't really know for sure until we try.
This was a popular area of questions, and for Dr. Benke, this sort of piggybacks on what Dr. Five Marisic just said. Does it make sense in your estimation to treat older patients with this approach? Can that progression be reversed?
Sorry, is this a question for me?
Yes, Dr. Benke.
Okay. Well, ideally, we would, I mean, the first thing is we want something that's safe. And the next thing was we want something that's effective as well. And so, you know, as Dr. Five Marisic was saying, you know, I think it all kind of depends on the animal studies about, you know, what we think is going to perhaps work in humans. But I think many of us think in the back of our minds, just because of what we know from neuroscience, is that there are these things called critical development periods. And so it might be better to treat earlier, but we don't know that yet. And so I think we're not going to know till we try. It is interesting that there's another company called Taysha. They've announced on their website that they're going to pursue a clinical trial of gene replacement therapy in Rett syndrome. So it's very specific for Rett syndrome, but they're going to start with older patients first before they pursue younger patients. So I think that that's interesting, and we will learn a lot from that study.
And Dr. Benke, maybe you could speak more. The next question I have here is asking about that older population. What maybe we've learned from some of these trials that are coming to clinic and protocol design, what it might look like for those older populations. That this parent asks about a 25-year-old girl with CDD.
We really don't know what the protocol is going to look like till we know what it looks like. That's not a very good answer, but it's true. You know, and the same goes for what the protocol is going to look like for the Taysha trial. I mean, nobody knows what that's going to look like till we know what it looks like. Yeah, I don't. So we just have to wait and see. But you know, I imagine that what they're going to do is, you know, that it's like any trial. There's going to be an exclusion and exclusion criteria, and they're going to be very specific about, you know, who gets treated first. They're going to have strict guidelines for that, kind of the same way that was done with the onasamnogene obopiravir for spinal muscular atrophy. You know, they will dose one patient in the whole country, and then they will wait a period of time before they dose the next one. It's, you know, safety is very key, and so the trials will go very slowly in that regard because of that safety issue.
Thank you. Dr. Five Marisach, this comes from a parent asking, I've heard that the CDKL5 gene is too big to put into common AAV vectors, but it seems you've accomplished that. Or is this just a small part of what's necessary?
Yeah, so I'm happy to be able to say it does fit. Our vector contains the entire coding sequence for the brain isoform of human CDKL5. So it's the whole thing. It's not too big, and it does fit into a single-stranded AAV9 vector.
Very exciting. And again, back to you, Dr. Benke. If I understand correctly, girls with CDD typically have 50% of their cells expressing the wild type CDKL5 gene. If this is true, why is 20% cell treatment with gene therapy adequate or clinically relevant?
That's a great question. It has a lot to do with the mysteries of what does CDKL5 actually do. You know, we think that it's involved in a lot of different circuits. So circuits are a bunch of neurons that are talking to each other specifically to perform a certain task. And how many neurons in that circuit do you need for that circuit to work good enough to either not have epilepsy or to have better motor function? So these are some of the mysteries of the brain that we still don't fully understand, as to, you know, how many neurons in a circuit do you need to work for these functions? And again, we don't know how much CDKL5 is necessary to improve the function and just say that percentage of neurons for things to work well enough. But you know, I think that is one of the take-home messages from Dr. Fife Marisich's talk is that we were surprised with just that many expressing there was improvement, not in everything, not completely back to normal, but there was improvement. And so I think that, you know, we have what we have in terms of AAV being the taxi that we have, and it only gets to, you know, 10 to 25% of the addresses, you know, neurons that it needs to get to, but it still seems to function as a good enough taxi to get the job done.
And reason for hope that little of expression is making an impact on a number of those behavioral tests that you were doing. So it is a reason to be excited. Dr. Lau, to extend that thought, the presentation paints a hopeful picture, almost as if the cure is already in hand, and that the only thing missing is the delivery trucks, as Dr. Benke mentioned. That seems like a problem that can be overcome. And we were told that a reasonable time horizon is 2 years. Is this really true? Or to put it bluntly, is this more of a marketing presentation from a pharmaceutical company? We are so new to this diagnosis, and the emotional shock is intense. I'm looking for hope, but I don't want to live in false hope either.
Thank you. This is difficult. We understand that the information discussed in these types of presentations does provide hope for families. But at the same time, it's important to acknowledge that there's a significant amount of work that still must be conducted. So before we can even develop a therapy, we need to understand the molecular basis and biochemical basis of the gene that's involved, of the disease, and how that disease changes over time. So for CDKL5 deficiency, that first step was identifying that gene over 15 years ago. And then we had to understand how CDKL5 functions, and we're learning about it. So there is progress being made. And this research has helped move the entire field significantly closer to the clinic. But you're right, it's always difficult to predict how long it will take from the start of preclinical work to enter the clinic or to begin conducting clinical trials in patients. Separately, the field of gene therapy itself has progressed forward enough that we are now able to easily determine the sequence of the gene of interest and then learn how to create the transgene and put it into the vector. So that has progressed forward. But that transition still is also very difficult to get into the clinical trials. And we have to rely on the success of the preclinical program in order to move forward. So once we decide to move forward to trials in humans, it'll take additional time. Why? We need to design the appropriate trial. How do we measure the right aspects of the disease so we see what is working? And then we have to analyze those results. That takes time. This is why it will take several years to get to clinic, actually. And we do recognize that this is difficult because while we are working to develop this potential therapy, you, the patients and families, are continually to be significantly impacted by this disease. So, it is difficult to give hope, but we don't want to give false hope.
Thank you, Dr. Lau. I appreciate that. I'm going to transition back to you, Dr. Benke. I think you really did a good job addressing this in your talk, but I will ask it again. And this is a tough one. What side effects can therapy have, and is there a downside?
I would say that what looks like the biggest side effect is the immune response, and we are, the AAV is not causing necessarily a problem. The body is doing what it's supposed to do. It sees something that it hasn't seen before, which is the AAV vector, and it's responding like a cold. It's trying to get rid of it. But we don't want the body to do that. We want as much virus to hang around in the body for as long as possible, delivering its payload to as many neurons as possible. And so what's going to be required is immune suppression. And there are side effects from immune suppression. If your immune system is suppressed, then you are more likely to get ill. And so there's going to be some issues associated with that. And the immune suppression medicines can have some side effects too. One of the aspects of delivering this directly into the central nervous system, we think that there will be less systemic side effects seen, like was seen when with the therapy for spinal muscular atrophy, which was given intravenous. And those side effects were liver irritation and some irritation to the blood. But we're hopeful that we won't see those, but it's absolutely something that will be monitored as part of safety with any trial. Do you think that answered the question there?
I think you answered as best you could.
Yeah.
That, like you said, we live by the protocol. We won't know how we'll best handle any of the immune system impacts until we get there.
I'll add a little bit. I think one of the questions is the vectors designed to express CDKL5 in neurons, and it's supposed to ideally not overshoot and express too much. But at least in terms of what we know in terms of humans, is there something where there are humans that have too much CDKL5? And there are some reports, but it's not clear that it's really necessarily disease causing at this point. So that's what we know.
Well, it can only, we're all learning on the go, I think, about what CDKL5 does. And as we learn, we'll use those to get closer to our goal that we're all partnering on. Dr. Fife-Marisic, I have a question that kind of harkens back to your original talk that you gave, that you were able to share more about your work in the microtubules and biomarker discovery. This one's long, so we can repeat it if we have to. But in the part on the microtubules, you are explaining that the dendrite appears, or this person's impression is it appears simply as a deflated balloon, which would simply be more inflated once we put CDKL5 back in, as if all the cellular mechanics remain intact, waiting for the missing protein. However, elsewhere in the body, an organ or muscle that isn't used will deteriorate over time. What do you know about how much of the healthy capacity of the fully grown dendrite remains dormant and come back to life upon successful protein expression? Are they fully or partially restored in any of the preclinical specimens?
That is a great question. Really, really in-depth question, but I love it. It's really, really
The
good.
parents are pretty sharp, I've got to tell you.
Very, very sharp. I 100% agree. So we did not examine dendritic spine morphology in our mouse study. So I can't answer that question specifically, but others have looked at this after using CDKL5 protein replacement therapy. And that's a published study it was published in 2018. Trazi et al. Is that study. And they did this in mice. And they did show improvements in those dendritic spines after putting the CDKL5 protein back, as well as behavioral improvements. And so in our study, we decided to look at behavioral improvements as sort of a surrogate for the changes in dendritic spines. And because we dosed the mice, later, but between the ages of about 4 to 11 years, human equivalent, it does suggest that even at those later stages that the brain has the capacity to recover. But we didn't look specifically at the dendritic spines. And then, we also, again, know that from Joe Zhao's recent paper, looking at behavior, similar to how we looked at behavior when he restored CDKL5 in older mice, he also saw behavioral improvements. So we think that there must be still some capacity for recovery there.
Well, and I think, as Tim said, that learning about the circuitry and how much we need and the time windows of when we need it, right? And what's still changing, and the neuromaintenance aspect that we've been hearing a little bit about, right, is all plays into that. And we still are sort of dissecting those aspects of CDKL5 deficiency disorder. So we all look forward to, I think, learning what role this all plays once we move from artificially turning on and off CDKL5 and replacing the gene the way you are, and what that'll do in different critical periods. Heather or Dr. Lau, if I may, what is the percentage of recovery of the child after gene therapy, and how quickly can the child recover? Are there any thoughts on this?
Thank you. It's really important to acknowledge that we have not studied our gene therapy in humans yet. And so we're still in the preclinical phase of research. So we don't know for sure how well a child might respond to a gene therapy because we haven't conducted the studies yet. So we do try to use our animal models to estimate what might be possible. And as Dr. Fife showed, we used our animal models to do this. And so our mouse model of CDKL5, we were able to fix some of the behavioral deficits, but not others. And we might see these improvements even a few months after treatment. So in humans, recovery may be variable and may impact only some of the symptoms of CDKL5 deficiency. So, and the response to treatment may not be immediately observed, and it may take time to see if our treatment is working or having a positive impact on some or all of the symptoms. Ideally, we would hope for reversal of all CDKL5 deficiency symptoms, but this may not be possible. So we need to study this further. We also have to make sure that it's safe for our patients.
That's become a common theme, that safety is key, in both preclinically and clinically, that we make sure we decipher that before anything else. Dr. Benke, this speaks a little bit to what Heather just spoke to, but with a twist, and it involves a little bit of speculation. Do you know which symptoms this therapy might help? Epilepsy, cognition, motor skills, would any of these be the symptoms you would anticipate having a change? Will gene therapy be recommended also for mild forms of the pathology?
It's hard to say what will improve. The preclinical data that Dr. Fife was showing suggests that from the organoid studies, it might improve epilepsy. the mouse model of CDD does not have epilepsy the same way that humans do. So it's not clear yet, and from the mouse model studies, how that would improve. But many of the aspects from the motor, how the mice move, and also how the mice learn in different tasks, those were both improved. So I'm hopeful that in human studies that you would see improvements globally. And this is one of the things that we've focused on when we're trying to develop outcome measures. We're not developing outcome measures just to count seizures. We want to be able to measure across the breadth of the individual what improved, and to be able to measure that, be able to, in a position to measure that as accurately as we can. So we're gearing up to measure everything that could potentially improve in CDD. That's the whole goal behind what we're doing.
And I heard, because I have the honor of working with our caregivers on the board, say, small changes make a big difference in their lives. So any meaningful change could, in those categories that you showed, is a big deal for their daily lives. Dr. Fife-Marisic, if the expectations of early successful deliveries of the healthy gene is that it would only be able to target some 10% of the neurons, is it reasonable to assume that as the technology advances, you can repeat similar treatments and activate more and more neurons over the years?
So right now, no one in the field of gene therapy can redose patients who have received AAV9 gene therapy. And this is, and we've talked a little bit about this today, really because of that immune response. So the antibodies, neutralizing antibodies that are generated would preclude a subsequent injection from being efficacious. So hopefully with advances in the field over time, changes in the way we can dose might become a possibility, but for now, we're only able to dose one time.
Thank you. And we're getting a lot of when, who, how questions. So Dr. Lau, when are the first test plans and how many people will be in that group?
Thank you. So we do not currently have a timeline for when the first clinical trial in patients living with CDD will begin. So because our research on CDD is still in the very early phases of development, it's going to take some time to develop plans for even creating that future clinical trial, including how many participants will be studied in the trials. It's also important to note that because we are in that very early preclinical stage of research and development, it is very possible that our early work may not successfully lead to the clinical trial, and that's difficult too. So that's all I can say about that at this point.
Dr. Fife-Marisic, this is, they're asking when would this be available, which is essentially the same question. And I imagine you're going to echo what Dr. Lau just said.
Yeah, I think I don't have much to add. We really hope that we'll get there, but we can't promise anything now. So it's just really too early to tell.
Dr. Lau, I have a few. I'm going to ask you one right after the other. I'll give you a chance to answer in between. If a patient from outside of the US comes to America and stays in the country for the duration of the study, would they be eligible to participate?
So again, we have not yet identified where we would conduct a CDD clinical study, correct? So it is, though, important to understand the geographic distribution of our patients living with CDD. It's also important to understand the requirements of local regulatory agencies in the individual countries to ascertain feasibility of conducting a clinical trial, okay? So a research study that's designed will define criteria for eligibility and patients are screened to determine if they meet this eligibility and then can be enrolled, right? So enrollment in the study also requires understanding the risk, the benefit, the potential requirements, such as participating in longer-term monitoring, undergoing the assessments and tests that are part of the trial, and a willingness to comply with the study design. So it's really hard to answer that at this stage.
Another question coming from the families is whether once we get to that stage, there may be a possibility of the participation of the families being paid.
Oh, right. So this is not an uncommon question and concern for patients and their caregivers and families. So again, we don't have any clinical trials for CDD deficiency. I have to say that at this time. Our research is still in the early phases. But as a rule of thumb, Ultragenyx does not pay caregivers or patients to participate in the clinical trial. However, we do provide reimbursement to cover the costs of reasonable travel and out-of-pocket costs that the caregivers and patients may incur while you participate in a trial by Ultragenyx. Okay.
Yes, I think that's fantastic. And then this, I mean, you guys talked about the pipeline of clinical trials and everything that needs to happen, and Tim reviewed where we are. This is kind of even on after we've had a successful trial, how we get into talking to payers and pricing and all of those logistics. They're asking about, will insurance cover any of the cost? And if not, what would treatment look like financially?
So that's for me to answer, but it's very difficult to answer that at this stage because our investigational product has not been studied in humans and it's not yet been approved by any regulatory authority. And that approval is first required prior to any consideration of reimbursement or pricing discussion. So that has to happen first. And I really can't comment further about the reimbursement after that.
I think the only example that's there is for families to investigate what's going on with onasamnogene albupervivac, and it's very different from and that's for SMA, and that's very different from one state to the next, because in many instances those individuals have Medicaid, and what Medicaid will reimburse is different from Texas versus Colorado versus the state of Washington.
So that is useful, and we'll try to make sure that those resources that you got in there about the SMA trials are shared with the caregivers so they can dig into that a little bit deeper. Dr. Fife-Marisic, this is an interesting one. Seeing the patient group of CDKL5 is so small, I imagine that relative research funding and resources dedicated to it are small in comparison with other more common genetic disorders. But I also imagine some of the gene therapy toolkits can benefit treatments across the spectrum. What other promising advancements regarding other disorders are of special interest in this regard? Is it reasonable to assume that if a delivery system or technique is successfully developed in another similar disease, it would apply here also? If so, which?
That's another really great question. So yes, absolutely. Learning from other studies as much as we can is the goal here. And we often look to the outcomes of research in other genetic disorders to gather insight in how to approach a disease and to learn what works and what doesn't work. Gene therapy is being applied to other neurological disorders now. And so we can start to learn about questions like, how do we best deliver the gene therapy? What's the most effective route? What type of viral vector is safe and effective in the central nervous system? We can learn about potential side effects of AAV9, and we've talked a lot about that. And we've had some great learnings in the field so far about the immune system, and how we can learn how to reduce the risk of side effects and enhance the delivery of the gene. So we're definitely paying attention to everything that's going on out there and learning as much as we can. The great thing is the same platform, right? AAV9 is being used for many other genes of interest. Each gene therapy is going to be a little bit different, but when the platform is the same, there are some learnings that we have. And, thinking about these things can help us avoid the same pitfalls and plan the best approach to test this out. And we can look at diseases like Tay-Sachs or SMA. We've talked quite a bit about that today, or other genetic disorders that are determining how to deliver the gene therapy to the brain. So, we're always thinking and learning, and we'll continue to do that. And that's the wonderful thing about gene therapy, particularly in the CNS. It's so new. Every day we're learning more and more. So we are watching and learning.
Thank you to all of you for, we've accumulated a few more questions in the QA, but I know you guys have strict regulations on what you can answer behind the scenes in the company. We'll keep track of them and submit them to you. I'm going to hand it over to Karen to kind of close us out. Thank you again for all of your presentations.
Thank you, Heidi, for doing the Q&A session. And we want to thank each of the panelists for their participation in this town hall. And we want to acknowledge the efforts from the patient advocacy program at Ultragenyx to help us pull this talk together. We also want to thank each of you for listening. Family support and education is a huge part of the mission of the IFCR, and we will continue to work to bring these types of opportunities to the community. As Heidi mentioned, we know some of you put questions in the Q&A chat box, and we will be distributing these to the panelists to get some answers. And also, if you are one of the participants listening to this later and you have a question, feel free to email that to science@cdkl5.com, and we will do our best to get these questions answered for you. We look forward to hearing from all of you, and we hope you all have a really great evening. We hope you feel like this was beneficial. I know I think it was. So much of this is hard to understand, and I want to thank all of the participants for helping take some really difficult information and answers that sometimes they can't even answer, and being honest and saying, we don't know yet, because that's what our